Hi sir, this is M. Mahesha from Triple E Department A section. My topic name is Principle and Applications of NMR Spectroscopy. Principle of NMR Spectroscopy. NMR spectroscopy is based on the principle of nuclear magnetic resonance, where nuclei with an odd number of protons and or neutrons have a magnetic moment. When placed in a magnetic field and exposed to radio frequency radiation, these nuclei can absorb energy and resonate at a specific frequency. The resonance frequency is determined by the strength of the magnetic field and the local chemical environment of the nucleus. The summary of main information we obtain from an NMR spectrum. Proton C and D are neighboring and split each other into two peaks. B. These protons B are split into four lines parted by the neighboring three protons A. These protons A are split into three lines tripled by the neighboring two protons B. First one, the functional groups that are present in the molecule. This is determined based on the positions ppm of the signals on the spectrum. Most often, the scale goes from 0 to 12 ppm. The number of protons represented by each signal is measured by the integration, which is the surface area under each signal peaks. S. Third one, the number of different types of protons in the molecule. This is determined by the number of NMR signals. Only non-equivalent protons give different signals chemically. Equivalent protons give one NMR signal regardless of their number. Fourth one, the spin splitting cell. How many protons are connected to the neighboring carbons? That is determined by the number of the peaks signal multiplicity within the signal based on the n+1 rule, n being the number of neighboring protons. NMR spectroscopy instrumentation. NMR spectrometers consists of a powerful magnet to create a homogeneous magnetic field, a radio frequency transmitter and receiver, and a computer for data analysis. Samples are placed in a tube and inserted into the magnet, where they align with the magnetic field. By varying the frequency of the radio frequency, radiation, different nuclei in the sample can be selectively excited for analysis. Third one, chemical shift in NMR. Chemical shift is a key parameter in NMR spectroscopy that reflects the electronic environment around the nucleus. It is expressed in parts per million ppm and is influenced by factors such as electronegativity, hybridization, and neighboring atoms. The chemical shift scale is referenced to a standard compound, typically tetramethylsilane TMS, which is assigned a chemical shift of 0 ppm. Fourth, spin coupling in NMR. Spin coupling, also known as J coupling, occurs when two nuclei interact through their magnetic fields. J coupling provides valuable information about the connectivity and spatial arrangement of atoms in a molecule. The number of peaks in a signal can indicate the number of neighboring nuclei with which a nucleus is coupled. 2 spin coupling, 3 spin coupling. When I and T are not equivalent and J1 not is equal to J2. When I and T are equivalent and J1 is equal to J2. Fifth one, applications of NMR in chemistry. NMR spectroscopy is widely used in chemistry for structural elucidation of organic compounds, determination of stereochemistry, and identification of unknown compounds. It is a powerful tool in drug discovery, environmental analysis, and quality control in various industries. NMR can also be used to study reaction kinetics, protein structure, and metabolic pathways in biological system. The summary of main information we obtain from an HNMR spectrum. Double, fourth double. Protons C and D are neighboring and split each other into two peaks. B. These protons B are split into four lines by the neighboring three protons A. These protons are split into three lines triplet by the neighboring two protons B. First point. The functional groups that are present in the molecule that is determined based on the positions of the signals on the spectrum. Most often the scale goes from 0 to 12 ppm. Sixth one, applications of NMR in medicine. In medicine, NMR spectroscopy is utilized for non-invasive imaging techniques such as magnetic resonance imaging for visualizing internal body structures. NMR based metabolomics is employed for disease diagnosis, monitoring treatment efficacy, and understanding metabolic pathways in living organisms. NMR is also used in pharmaceutical search for drug development and formulation studies. Application of NMR in medicine. Clinical application of proton imaging in diagnosis. Brain. Distinguish grey matter and white matter. Imaging posterior fossa, brainstem, spinal cord. Detect demyelinating lesions, tumors, hemorrhages, infiltrations. Seventh one, advantages of NMR spectroscopy. NMR spectroscopy provides detailed structural information without the need for extensive sample for preparation. It is highly versatile, non-destructive, and can be applied to a wide range of molecules, from small organic compounds to large biomolecules. NMR experiments can be tailored to specific research questions, making it a valuable tool in various scientific disciplines. Eighth one, challenges in NMR spectroscopy. NMR spectroscopy requires specialized expertise and expensive instrumentation, which may limit its accessibility to some researchers. Signal overlap and low sensitivity can pose challenges in the analysis of complex mixtures or dilute samples. Careful data processing and interpretation are essential to extract meaningful information from NMR spectra. Eighth one, future developments in NMR spectroscopy. Advantages in NMR hardware such as higher magnetic field strengths and cyanogenic probes are improving sensitivity and resolution. Computational methods for spectral analysis and structural prediction are enhancing the capabilities of NMR spectroscopy. Integration of NMR with other analytical techniques, such as mass spectrometry and X-ray crystallography, is opening new avenues for interdisciplinary research. 10th one, conclusion. NMR spectroscopy is a powerful analytical technique that provides valuable insights into the structure, dynamics, and interactions of molecules. Its applications span a wide range of scientific fields, from chemistry and biology to medicine and material science. Continued advancements in NMR technology and methodology will further expand its utility in research and innovation. Thank you, sir.